All right, everyone. Welcome to the 2024 AGM Preclinical Translational Science webinar series. We're very excited for you all to be here. I'm Serene Marcosian. I lead the Assay Guidance program here at the National Center for Advancing Translational Sciences. So this season's first webinar is by Dr. Derek Lowe. We're really excited to have him here today. He's the Director of Chemical Biology at Novartis. He received his PhD from Duke in 1988 and then did a Humboldt Fellowship in Germany. Since 1989, he has worked at several different biopharma companies in a variety of therapeutic areas, including CNS, metabolics, anti-infectives, oncology, and many more. In the last 10-15 years, his research work has shifted more to chemical biology approaches to better assess target selection and other fundamental problems of drug discovery. And as many of you already probably know, he's the author since 2002 of the very popular blog In the Pipeline, which covers a variety of chemistry and biology subjects in what is now likely the longest running site of its kind. We're super excited to have you here, Derek. Derek's going to tell us more about AI in drug discovery, when, where, and how much. And just to note that we are happy to take all of your questions and discuss the questions at the end. So please type in your questions in the Q&A box, and I'll moderate the Q&A session at the end of the talk. Thank you so much again. And, the floor is yours. We're excited to hear your thoughts.
Thanks very much. I mean, that's a nice intro, and it's true. I have been turning into more of a chemical biologist over the years. I'm still a knuckle-dragging organic chemist at heart. That's where my interests always come back to. But I find that's very good to have that background as you go off into the more biological topics, because it gives you a point of view that not everyone has. Not necessarily a useful point of view, but it is a point of view. So, to talk about AI and ML and all the modern era, I'm going to go back for a slide or two to the pre-modern era, which is when I started in the industry, 1989. And the view from there is very different. And over the years, I've been realizing just how different it is. And now it's starting to look like some sort of weird foreign country or movie set. No email, no web. Desktop computers were rare. The company I work with was adamantly opposed to putting computers on scientists' desks. Overhead transparencies for all presentations, slide projectors with real slides and real bulbs that burned out. Medicinal chemistry was very different. We didn't have really any metal catalyzed couplings going. CombiChem was just a gleam in a few people's eyes. There was not much in the way of analog by catalog. Those of you who do chemistry know now that you say, oh, well, I'm just going to make 300 amides. Let me order 300 diverse amines from all these companies who want to sell me chunks of them. None of that stuff going on. And we ordered our chemicals by filling out a 3-layer multicolored carbon paper form, which really seems like about one step from writing the order down with a sharp stick on a buffalo dung tablet and drying it in the sun. And I'm glad we don't do it that way anymore. And I'm also very glad we don't do literature searches the way we used to. Online searches were generally done by some sort of library staff. PubMed didn't exist yet. Bound volumes of journals, bound volumes of chem abstracts. And if you wanted to search through them, you did a lot of page flipping and volume turning. And you took them to the photocopier and did a flip and turn move that people from that era will remember and no one else has fortunately had to see. But a revolution was already underway, even at this point. And I got to hear about it as soon as I joined, because I was in a CNS project working on muscarinic receptors. And everyone was very excited because we were getting away from tissue slice assays. That was the standard in the muscarinic literature. And for example, there's a couple of papers from back in the day. And this is going to sound pretty primitive for people who came into the field decades later. But this was the standard. Rat vas deferens, possum trachea, all sorts of stuff like that. And that's how you assayed your compounds, because these tissues were more or less enriched in more or less the receptor subtypes that you thought of. Well, we were very happy to get away from this primitive Dr. Frankenstein stuff and move expressed purified membrane preps of individual receptors. And we thought that is really going to help us sort things out. more about that in a sec. What it also meant was that high throughput screening was now becoming really possible. And that made people wonder, what are we going to screen when we run out of compounds? My gosh, these assays, they're so amazing. We're going to zip through everything we have. And the answer, of course, was the rise of CombiChem, cranking out more compounds than the robots could screen, which made people ask, how are we going to screen this stuff? So there was an arms race going on. And here's a 1997 paper talking about that. How are we going to improve the assay techniques to deal with this flood of amazing CombiChem? Well, that flood turned out not to be so amazing. And now I think the assay throughput is definitely in the lead, except we now have screening technologies like DNA encoded libraries where you have potentially billions of compounds per little Eppendorf vial. So you might say that the compound numbers have definitively won in the end. But I think we reached a détente, an understanding. Problem is, all this CombiChem revolution led to trouble. The chemistries that were developed for this tended to make a lot of really similar looking compounds. And they tended to be compounds that did not match up with drug discovery as well as we'd hoped. This is a review from my old colleague, Pat Walters, when he and I were both at Vertex, and he goes into great detail on this. It really was a disappointment. People were frantically screening all these amazing piles of CombiChem compounds and getting far lower hit rates than they ever expected to get. So we ended up with what I have, and you'll see this theme recurring as I go on the talk, what I think is a problem of reductionism. This is Pat again, saying that a lot of these trends accelerated along with advances in molecular biology. Understanding these receptor subtypes, being able to screen them as purified membrane preps, a lot of more target-focused drug design as opposed to the older phenotypic stuff and tissue-based, which is like one step off phenotypic. And you ended up optimizing a lot of parameters without really getting them into cells, tissues, and animals as early as they should have been. And we also managed to find all these metal catalyzed couplings that we lacked, which allowed us to stitch together ugly, not very soluble molecules more quickly than we ever had before. So reductionist models led us down the garden path. I think that that entire era, in retrospect, is going to look like something of a mistake, at least something of a dead end. And this is a quote from a paper by the pharma consultant Jack Scannell, very interesting paper that I'll come back to as well. He points out that we were exhausting models that were predictive of clinical utility. That meant we were having to go to models that didn't have very good predictive value. And that is because good reductionist models are difficult to find. So you use bad ones instead. You have to do something. The moral hazard, though, here is that it's easy to do that and still pretend that you're being productive and still tell upper management that you're being productive because you're prosecuting targets, you're running assays, you're running screens, you're advancing things to the clinic. But what you don't see underneath is the rot of predictive value. And I'm hoping that we're getting out of that through several revolutions. One of them is the chemical biology revolution, which has, of course, grown into a very large field, and it's sort of penetrating all of drug discovery and a lot of academic science as well. We have all these chem bio techniques and molecular biology techniques that are so useful and so powerful, they're just widely adopted. A second revolution is structural biology. X-ray, of course, has been getting faster and more automated all the time, but the cryo-EM revolution has really shaken things up, giving a structure to things that you never, ever dreamed you'd see structures of. And, of course, the recent computational advances in protein structure, which we're about to talk about, really have shaken things up. So is AI and ML, I'm going to just conflate these from now on, computational techniques, is that the third revolution? That depends on what you're trying to improve and what your expectations are. So my own view is that the AlphaFold, Rosetta Fold, I'm not going to, put David Baker in second place. My own view of that is that approach is a major achievement. It really is. It's stunning how well it works when you think about it. But I don't believe that some and reason is that a lot of our fundamental problems don't have very much to do with protein structure to start with. And when I say that, some people in the audience nod their heads, yep, yeah, I hate to say it, but he's right. And others are just looking at me like, you know, I'm drunk or something because it doesn't seem right. But it's true. Our biggest problems don't have to do with protein structure. Here are what some of these problems are like. This is what a medicinal chemist's problems are. You have what the immediate problems right in front of you. What compound should I make next? How should I make them? These are chemistry problems, medchem problems. What analog should I do? Should I put nitrogen here? Should I put this chiral center? Should I make this in more electron rich, electron poor? What should I do? There's so many possibilities. And once I've settled on that, how do I do that? What does the literature tell me? What's the fastest, easiest, cheapest, most simple-minded way to make it possible? Because that's what we're looking for. It's, as an aside, a lot of people who come into medchem from very high-powered organic synthesis groups are in for a rude shock because they've been used to troubleshooting the retro-Zamboni rearrangement at step 38 on 0.67 mgs of material. And they get into the medchem labs and they find they're banging out amides and palladium couplings, a bunch of stuff that's from the first semester of sophomore organic chemistry. And it can be humbling until you realize that we're not in the ars gratia artis field, the art for art's sake. Chemistry is not its own reward. It's a tool in the box. And frankly, if the drug companies could find ways to make all these compounds without employing a bunch of chemists, they'd fire us all. Fortunately, they haven't. So these are the immediate problems. The bigger problems are, how potent and selective do these compounds actually have to be on this project? How do we know that we've reached the zone that we need to be in and we can stop, worrying about those and go on to something else, such as balancing those properties with the PK and metabolism properties that you need for dosing, preferably daily dosing, orally, etc. What counter screens and tox do you have to worry about? What can we do to minimize our risk as we go on? Very tough questions, but essential to any medchem project. And then you got the big one. What project should we even be working on? Is this the one? Should we be doing this project at all? Is there a better use of our time and money and effort? Should we be doing this therapeutic area at all? Is there a better use of our time, money, and effort? Now, this is a really strategic level problem. And what I would like to say is that the ability of AI and ML techniques decreases drastically as you go up this hierarchy. Drastically, because these problems become much more wide-ranging and complex and full of knowledge that we don't necessarily have. It's kind of like a gigantic hand of 500 card stud poker with 1,000 people sitting around the table. You see some cards, you don't see others. And when you see some cards, you wonder, is that what they want me to see? Or is this something they had to show me? How do I make decisions based on limited knowledge? AI and ML are very, very poor at making decisions on limited knowledge. What they are very, very good at is rearranging existing knowledge. So let me take a second to illustrate what I think is the drug industry's fundamental problem. And this is what people really would like for artificial intelligence to be able to help us with. If you just go around here, there we go, that 13.8, that is the overall success rate from entering the clinic to getting to an FDA submission. Over 85% of the projects fail. And I would like to advance the idea that, and it's not original with me, that we are the only major industry that works under these conditions. I mean, you'd have to think that 85% of Honda's cars are capable of rolling out of the assembly plant. 85% of Pizza Hut's pizzas are as good as they're ever going to be. But 85% of our drugs fail in the most late stage, painful, expensive, time-wasting ways possible. And if there were any way for us not to do that, we would love to do it. There are tens, hundreds of billions of dollars piled up out there waiting for you to scoop them up with a front loader if you involve even a small degree. And the fact that it hasn't been solved tells you that it's probably a pretty hard problem because the incentives are there. So let me get back to AlphaFold and illustrate what I'm talking about, rearranging existing knowledge. Many of you may be familiar with John Searle's philosophical thought experiment from the early 80s, the Chinese room. He said, and I believe that is actually a character of John Searle himself, he said that if you put him in a room, or some lucky person in a room, with a gigantic set of rule books about the Chinese language, huge amounts of data to sort through, then if someone dropped a question written in Chinese through a slot in the wall, he could sit down and start applying all these heuristics and say, aha, he doesn't know Chinese. Let's make that clear. He never learned it. Okay, this symbol that has two little crosshatches and a downward stroke with a curl and above a such and such with two dots, that generally means you have to respond with something like etc. And if you go through that in enough painstaking, ridiculous, inhuman detail, you can generate something that looks like a meaningful response. This is what ChatGPT does. This is exactly what ChatGPT does. It is Searle's Chinese room instantiated. That's what a large language model is. It looks through vast amounts of human writing and human speech and says, oh, okay, when this particular pattern of words shows up, then often people answer with patterns of words that look like this. It is a plausible sentence generator, and that is what Searle is working on doing, is a plausible sentence generator in Chinese. He doesn't understand Chinese any more than ChatGPT understands English or whatever symbology you're using to feed it. So that's exactly what AlphaFold did with protein structure. It is a pattern matcher. It doesn't embed any of the thermodynamics, any of the stereoelectronics behind protein structure. It just says, okay, when you have glue, veil, lyse, tire, okay, that generally makes a loop over like this. And then if there's a pro, it shifts it over like this, and then and it works startlingly well. But my feeling is that if protein structure was a problem put to the human race by some supreme being to test us, we flunked, because we do not still understand why proteins fold the way they do. We understand a fair amount of it, but we really can't do it from first principles. We cheated. We pattern matched our way to the solution. We shortcutted it. And now that's great for drug discovery in the same way that the chemists want to use these cheapo, quick, fast reactions to make drugs. We want the cheap, quick, fast way to predict proteins rather than the deep understanding Zen way of predicting protein structure. Here's an example from my own field, retrosynthesis. A few years ago, I was a lot more optimistic about this than I am now. And this is a quote from Boris Grabowski, who's a pioneer in the field back in 26, and talking about kind of channeling Winston Churchill, that is the end of the beginning, that you will turn these programs loose on the synthetic organic chemistry literature, and they will tell you very nice routes of how to make more complex molecules, because this looks kind of like this thing that was made in 2003, and that can be bought from this catalog, and that can be combined in this reaction that's been run since the 70s to do this, which should work with it, et cetera, et cetera. That's what we do in our heads, organic chemists. That's one of the things that synthetic organic chemists feel is their job, is to look at whiteboards and go, okay, I could make this like that, and if only I had that, but then, and make a big decision tree. Software was looking for a little while like it was going to take that away from us. Well, it may well, but that revolution has been delayed, because it turns out the organic chemistry literature, that data set, is in worse shape than anyone had feared. And that's saying a lot, because we already knew what the organic chemistry literature was like, and it's pretty shaggy. There's a lot of bullshit in there. There are a lot of reactions that don't work. There are a lot of reactions that didn't work. They're reported not to work that probably did, but got messed up somehow. You know, a little water got into them. They didn't work because they can't work. They didn't work because someone was a little sloppy. And one of the big problems is that there is a terrible lack of data. by negative data, I don't mean, stuff that didn't work that should have. I mean reactions that were set up properly and really didn't work. Well, we don't publish most of that stuff. No one does. We never have. But that's what a machine learning model desperately needs, is quality negative data along with a big range of quality positive data. That's what it sinks its little electronic teeth into. And we don't have it. There are also, as Grzybalski himself has pointed out more recently, a lot of silent biases in there. This metal catalyzed coupling reaction, that catalyst wasn't the thought of a lot of careful rationales and experimentation. Now, that catalyst was because it was cheap, or that's what was on the shelf, or that's what my buddy down the hall ordered and we had it and the other stuff got back ordered. The literature is shot through with a lot of silent, unconscious biases like this that make it less reliable than it should be. Grzybalski himself is saying that we may need to recapitulate a lot of the synthetic organic chemistry literature under controlled conditions, i.e. automated conditions, to get a data set that's really fit for a machine learning model to dig into. In other words, we've let down the side. We have disappointed the software by our human frailties, not for the first time and not for the last. So one of the things about these tools, which is both good and bad, is as the tools of both experimental chem bio and now computational tools become easier, it's getting easier to do it in a way. It's getting easier to do what used to be the standard, and you move on to harder problems. Freeman Dyson was fond of saying that new directions in science are launched by new tools much more often than by new concepts. If he's right, and he may well be, Dyson was tough to bet against in topics like this, then this is good news because we have a lot of new tools, and these may well launch us in a lot of new research directions. But on the negative side, lower barriers to entry means it's a lot easier to produce a lot of low quality work. And frankly, if you go to Midland or below chem bio type conferences, you will see this in action. You will see a lot of stuff done that you're like, yep, that is indeed a proteomics experiment. I don't know why you did it. I don't know what it tells us, but there is no denying it, that there's proteomics. So there is a lot of stuff like that being done, and that happens in every field as new tools come in. It launches some people into new areas of research, and it launches some people into a frenzy of hack work. A bigger problem, though, is that we run the risk, again, of getting seduced by detail. So let me tell you what I mean by that. Detail, of course, is a feature of drug discovery and molecular and cellular biology. There is an absolutely staggering level of fine detail. And of course, this is just one little slice out of that huge Roche pathways wall chart, which of course itself doesn't even begin to encompass everything that's going on, but it's enough to make anyone sit down for a while. And we have made huge advances by studying in great detail all these intricate tiny pieces, but we have paid a price for it. Reductionism is a tool. It's a great tool, but it's not the only one that in the box. For instance, take a look at this. All right, if that's a data set, how are we going to extract something interesting out of it? Well, we could run a bunch of XY plots and see how many different shades of color are in that. What's their distribution according to on the XY plane? Is there some sort of pattern in there? How about the size of each one or the shape? Some of them are more oval, some of them are like lines, some of them are perfect circles. There's got to be a lot of things you could understand if you would just really tease out all these data points. But you would never realize that that is a tiny part of this. That is a little bit of the skirt of this woman sitting in the shade on La Grande Jatte by Seurat. And if you go to the Art Institute of Chicago, which I highly recommend, you can see the original. And I don't know if they'll let you put your nose up close enough to it to see this, but trust me, that's exactly what it looks like. You cannot go from this to this. You can't go from this to her skirt. You don't know what it is. There's not enough data here to tell you that. And it is not something that you can run in reverse from reductionism. It's an emergent property. And that is why you cannot go from this to that. The It is an emergent property, and reductionism will only take you so far. Now, we haven't finished riding that horse. There's a lot of distance still to go and a lot of neat things still to be found, but we're going to have to come to terms with the fact that there are a lot of important things that we will miss if we only concentrate on the tiny details. So AI and ML, I think, are very fit for the reductionist approach. In fact, a machine learning program would have a great time with that little swatch of that Cyril painting. A machine learning program would rip through that and spit you out all sorts of correlations about the colors and the shapes and the sizes and the positions. But it couldn't go any further. And that is why I was showing that hierarchy of problems earlier. I think the higher level of the problem, the worse the traction is for machine learning and AI. And that's partly because of the lack of high quality data. That's why I was talking about the organic chemistry data set. We don't have enough positive or negative data to feed into the software. And that's partly due to the poor shape of our existing data. And it is especially due, I submit, to the size of those data sets compared to the problems. For example, you would love to be able to use something like AlphaFold to come up with ligands for the binding sites inside these proteins, or to predict what these ligands might look like. To do that, though, you would need a large data set similar to the large data set that informed AlphaFold and RosettaFold, that is to say, the PDB. There aren't many data sets out there like the PDB in terms of their coverage and especially their quality. Very, very well curated. Now, there are some weird things in there, but compared to a lot of other data sets, it's pretty pristine. I also think that the number of interactions that protein residues can make with themselves is smaller than the number of interactions that human-made artificial drug molecules can make with protein backbones. We put in things like fluorines and functional groups and heterocycles that do not exist in the protein world. So that means not only do you need something the size of the PDB, you need something much larger. But in the PDB, only 5 or 10% of those structures have any kind of ligand in them. And some of those ligands are things like, tartaric acid, which doesn't do you a hell of a lot of good. So we are extremely short of the data set we would need to do a machine learning AlphaFold, RosettaFold type initiative for small molecule drug discovery. Now, there's no fundamental reason that machine-based approaches can't help. That's the thing I like about them. There's nothing in physics or math or logic that says that you couldn't do this. It's just going to be a hell of a hard problem, and it could be quite some time before they reach their potential. What we need, and this is getting back to that 13% success rate, or more appositely, an 87% failure rate, God help us. What we need is predictive validity. And this is from that same Jack Scannell paper I mentioned earlier. And he's looking at high predictive validity over here. That's where you don't have too many hits, but they're all true positives. At lower and lower predictive validity, you end up with more false negatives and more false positives, and it gets hard. Here you go. Here's predictive validity low. Your number of false negatives and false positives overwhelms the number of true positives, and you have a much, much harder situation to work with. The true positives outnumber the crap if you have a great predictive validity. Otherwise, the crap outnumbers the true positives. So, problem is, we find ourselves mostly using low predictive value assays because the high predictive value ones we have already beaten to death. We already, for the most part, have worked those pretty hard, and there are pretty good existing drugs for many of these. So we need new ones, but that's not easy. The other depressing thing about predictive validity is, I'm not going to go deep into this graph. You have to read Scannell's paper pretty closely. But what it basically is telling us is that this PPV is another abbreviation, positive predictive value, the proportion of decisions that give you positive readouts. Note that this is a log scale here, which is kind of terrifying. And what he's saying is that if you do predictive validity and look at the log scale of throughput, it tells you that if you are in a bad a low predictive validity assay, it does not matter how high your throughput gets. You are still in bad territory. You cannot screen your way past a bad assay. This is the moral hazard again, too, because it's easy if you're running the screening lab, say, oh, we ran so many more screens this year. We screened so many more compounds, so many more diverse protein types. Give us a raise. Give us a raise. We need more money. But if this is all you're doing is screening into a void, then it's not doing anyone any good. Meanwhile, over at high predictive value, even modest increases in throughput pay off big. So you need high predictive value assays, but telling upper management that it may be a losing game and trying to explain how you're going to get those versus how you're just going to make the robot screen faster is definitely going to be a losing game for your performance review. Overall, there has to be a chain of translatability. There needs to be a shared mechanistic basis for the assay readout, the disease model, and the real human biology of that disease. That's applying predictive value, not just to an assay, but to the whole screening cascade. If we were better at this, we wouldn't have that 87% failure rate. But the fact that we do, after all these years and all this effort, and God knows all this money, tells you that getting better predictive value is not easy at all. But the goal is the assay endpoints and disease endpoints have to be as close as possible. So you can remember Einstein's advice, the theory has to be as simple as possible, but not any simpler. We have focused on breaking drug discovery down into simpler steps, and it made heuristic sense, financial sense, and a lot of scientific sense, but it's not going to go on that way forever. We are dealing with systems that are more than sums of their parts. So we have to be ready to ditch reductionism when we need to. Right now, I think AI and ML are actually fuel for the reductionist approach. And I think they're going to help us ride that even harder and further, and maybe help us come to the end of it a little faster than we would have otherwise. And it's going to be fascinating to see what comes next after that. Okay, that's what I have for you, and I'll take questions, and I'm sure there will probably be a couple after all that stuff. Thank you.
Thank you so much, Derek. These are excellent points of I'm trying to digest everything you said. Okay, so there are 2 questions in the chat. One is a comment, I believe. There's another one. And then we're open for questions, and we have plenty of time for discussion, people. So ask your question.
I want to leave some open for that, because I find when I talk about these subjects, it generally sets people off. So don't be shy.
Okay. All right. We'll start with a comment from our scientific director, and he's saying, Matt Hall, and he's saying, Derek had an opportunity to connect to Ferris Bueller here with the Syrah.
Yeah, it's true. It's true. That's a Chicago film all the way. So I don't know if I'm going to take that opportunity, but yeah, I know what you're saying. I remember that scene. Yeah, I was looking for an impressionist painting to illustrate that point, and I saw that and I said, that's it. Look at this. This just looks like a data set when you zoom in on it, but it's a lot more than that.
I actually have seen it in person in Chicago. It's gorgeous. So
it is.
Okay, so let's go to more questions now. So here's a highly ranked question on the Q&A box. Right. What strategies do you recommend for obtaining the most accurate model when solely relying on a homology model in the absence of a crystal structure?
Yeah, and of course, that's what people find themselves using these proteins prediction techniques for. I think a recent paper by the folks at Schrödinger present one way of doing it. They, of course, and if you know Schrödinger, you're not going to be surprised to hear that they're beating the drums for the FEP, free energy perturbation computational model. But they believe that by applying that with some experimental data to your homology model, that you can actually have a better chance of selecting the most relevant model out of what would otherwise be an inadequate data set to pick from. They just published something on this in Cell. I blogged about it a few weeks ago, and they may be right. I mean, of course, it's impossible to evaluate They show 3 examples where it seems to work, but there may be others where it doesn't. But they may have a point there in that you may be able to use a surprisingly short list of compounds with the actual experimental data. And as they emphasize, you really do need at least a little bit of ground truth experimental data in order to rank these different homology models. And you may have several different ones, all look equally plausible, in order to rank them into the ones that have legs and the ones that don't. So that would be my first suggestion. And it unfortunately does involve getting some experimental data. And you would hope to be able to compute your way to nirvana without actually having to get your hands dirty like that, but we cannot do it yet. People have been promising that for decades. There's a famous Fortune magazine cover story from the mid-'80s showing a bunch of folks looking at a very primitive wireframe molecule of a double helix, and this supposedly had something to do with the imminent drug discovery revolution. We're still trying to get there, and I don't think we're there yet. And as I mentioned earlier, I don't think that the large machine learning models are going to get us there because there's not enough data to feed into it. So I think that Schrodinger, whether or not you believe in FEP as the solution, I think Schrodinger is correct in that you're going to have to mix some experimental data into your approach to have any hope of picking the right one, because I don't think you can do it de novo computationally.
Thank you, Derek. There's plenty of questions that came in, so let's go through them. So the next one here is, can you share some interesting examples or stories of assays that you have seen do a good job of demonstrating high predictive validity?
Varies by therapeutic area. The therapeutic area that I think actually has the highest predictive validity is probably infectious disease, honestly, because if you can shut down a bacterial or viral infection in a cell assay, you have a much better than average chance of being able to do it in an animal once you've understood the PK of your compounds. And if you can get a compound that shuts down a bacterial or viral infection in a reasonably relevant animal, and there are only a few to pick from, and sometimes we're more like rats and sometimes we're more like ferrets, it just depends on the disease. But if you can shut that down in an animal, your chances of being able to shut it down in humans are very high. And that shows actually in the success rates back in that slide. The highest success rate is actually for vaccines. Only about 65% of vaccine trials fail. That is really the best in the industry right there. And it shows you what state we're in, but that is the best in the industry. And infectious disease is up there pretty high as well. And of course, at the bottom is CNS, where we have no understanding and no decent models in almost every case. So I would say infectious disease. Outside of that, I would say diabetes. The animal models for diabetes, and I'm talking about DB, and OB mice, are not perfect, but they're pretty good. And if you really can normalize metabolic behavior in one of those, chances are higher, much higher than usual, that you can probably do it in humans. Now, there are going to be other concerns, which is why just the efficacy model is not the only thing, because for diabetes, of course, you need vast amounts of cardiovascular safety. The FDA will not let you proceed without it because people are going to be taking this the rest of their lives, and your patient population is at risk for cardiovascular trouble to start with. That, as the DB mouse, is not going to inform you about that. But as far as mechanistically letting you know you're on the right track, it's pretty good. So those two I would rank high.
Interesting. Thanks, Eric. I wonder why some therapeutic areas and vaccines end up having better, more robust assays, more predictive assays than others. Is it because there's more work done or better understanding of the mechanisms of the disease?
Yeah, mechanistic understanding might be part of it. I mean, look at the amount of work that's gone into an HIV vaccine, and that has just been such a hard nut to crack. And then and some of these you can see why it's tough, because HIV, of course, because of its actions on the immune system itself, make it intrinsically difficult. And then you have things like malaria, which, because of their extreme ability to shapeshift all their little coat proteins and antigens, are tough. That's why you don't have a universal flu vaccine, because the flu is out there flu vaccine trading card meet every season. So these things mechanistically are not too hard to understand, but overall, the vaccine success rate is actually exemplary.
Thank you, Derek. All right, next question here, highly rated. What is a medicinal chemist's intuition and how can you model it?
Oh yeah. See, this is a good question because we mid-chem types believe that we have intuition and we believe it's pretty valuable stuff and we deserve to be paid handsomely for it. I have been gradually having that balloon deflate on me. I still put my oar in about various compounds, especially ones I think are really awful. But I mean, look at metformin. Metformin is a terrific drug, and it has been on the market for decades, and it really does a very good job on metabolic disease and type 2 diabetes, and we don't know how it works. We're getting closer. There are a lot of very good theories. We're getting closer, but for years, I mean, it was on the market with no one having a clue of how it works. Structurally, if you look at metformin through the eyes of a medicinal chemist, you burst out laughing. And then you turn red with embarrassment because it's the kind of structure that any med-chem person in their first years in the industry would immediately cross off a screening list. Oh, come on, you gotta be kidding. Metformin looks like the kind of thing you put into marine paint to keep barnacles from sticking to ships. It is a joke of a compound from a structural point of view. It also looks like it might blow up if you banged it with a hammer. It has so many nitrogens. So it's a standing rebuke to med-chem intuition because it goes against everything that we think is drug-like, but it has been a fantastic drug. So med-chem intuition can only take you so far. One other thing about that is some years ago, there was an effort by one of the big pharma companies to run lists of compounds past their medicinal chemists, their experienced ones, and say, which one of these should we get for the screening collection? One of the things they did, though, was as the weeks went on, they ran the same list of compounds back to the same people without telling them, and people scored them differently. The same chemist, looking at the same list of compounds a couple of months apart, ranked the compounds differently. So that is also a bit of a head scratcher when you start talking about med-chem intuition. It's not that it doesn't exist, but it's not always right, and it's changeable.
Thank you, Derek. Multiple questions. I'm going through the ones that are ranked higher than others, so hopefully we'll go through most of them. Okay,
Keep on.
Okay, thanks, Derek. I appreciate it. Let's see. This is I missed one. Okay, so reaction enumeration is small molecule ligand design in the probably one way where AI can be useful to generate ideas on ligand design. Not sure exactly. There's a question there.
Yeah,
I'm not
I
sure
mean,
if
this gets into the whole generative chemistry idea. Can you use AI type systems as idea generators? And I think you can, and a lot of companies, including the one that pays my salary, are working on that, but I haven't seen any great successes from it yet. And I would really love to see the best generative chemistry programs run in a head-to-head double-blinded competition with a room full of medicinal chemists and a coffee machine and a tray of donuts and see which one comes out ahead.
Thanks, Derek. I think there are some follow-ups on some of your answers there. So perhaps after the talk, you can go through them. I
Sure.
moved them into the answers. So I apologize for the one not to follow on your questions. All right, here's another one. How would you use AI to improve the efficiency and lower the cost of the drug discovery process?
I would not tell people that that's what I'm trying to do. That would be my first advice, because I have seen a lot of AI companies come through the doors over at more than one company, but especially the last few years. And so help me, almost every one of them, their slide deck starts out by basically saying, not quite in these words, but this is the message: comrades, the revolution is here. It's happening right now, and you better get on board. Unless, of course, you just want to sit there with your feet in the mud watching all your competition ascend into the sky to artificial intelligence nirvana. If you want to do that, you can, but I think you should sign with us. Honestly, that's what a lot of the slides come a lot of the press releases and a lot of the hype is talking about how AI is going to improve the efficiency of drug discovery. But for the reasons that I talk about, the things that, I mean, look at the reasons we have that 87% failure rate. It's mostly due to picking the wrong target in phase 2. And the rest of them are mostly due to unexpected tox in phase 2 and phase 3. And I don't see how AI is going to help with either one of those. We don't know enough to pick better targets, and AI is not going to produce something from nothing. AI rearranges the existing knowledge base you have. That existing knowledge base we already know to be inadequate, especially about toxicology. We don't know crap about tox. So I think that if you're going to deploy AI and ML in drug discovery, you are better serving yourself and your bosses by being more realistic about what it can do. And don't talk about improving the efficiency of the whole process, because that's an extremely difficult thing to promise. Right? Actually, it's an easy thing to promise. It's an extremely difficult thing to deliver.
That makes total sense. Okay, next highly rated question. Okay, so wonderful presentation. I agree with your statement on lack or negative results in chemistry.
Yeah.
With a race to publish in high-profile journals, how can we solve this question? Do we need a journal of failed experiments?
Yeah, people have tried to launch journals or data repositories like that over the years, and they have never taken off. We're going to have to change our attitudes, and that's going to be a slow process. I mean, every time I see something that the key step is, and then people are going to have to do something different than they've always done, or they're going to have to act differently than people act, I just despair, because that's a tough ask. But we really are going to have to change our attitudes about that. And I think that machine learning revolution may be a strong enough stimulus for us to do it. The other thing that could help is the fact that supporting information in journals is now effectively unlimited. I mean, for a while you had people complaining, oh, what's this 100-page SI bullshit? I celebrate when I see that. That means people were giving freely of their knowledge and their data. And that, I think, would be a good place for the negative results to go. Just put in a big old mass of all the stuff you tried. Throw it in there. Why not? It's all just electrons being stored on a server somewhere. You don't have to read it. The machines can go dig through it and see if they can find something interesting in there. So that would be my advice, is journals need to loosen up and allow people to talk about the stuff that didn't work and just stick it in the gigantic SI files. That's its natural home. And of course, authors are going to have to be willing to put it in there. That's a tough one, because we like to pretend that everything worked just the way our mighty brains worked it out in flashes of brilliant inspiration. It doesn't happen that way. You've been on projects and you've seen the actual papers that get published and the talks that get given. They aren't the way things happen. They never are. We should maybe stop pretending a little bit. But again, that's a hard ask. That's a human behavior problem.
Thank you, Derek. And at least the AGM program, we hope we are trying to help parts of that by bringing best practices for asset development, data analysis tools, as well as, providing folks with practical advice and bringing all these experts to share things like you are sharing, to, highlight issues that the scientists, get in the field.
You have to do that. And inside the pharmaceutical companies, there have been all these big initiatives to improve data quality. It's been tough. The data quality, I don't think, have been improving at the rate that we would like or the rate that we need. But the beatings are continuing until morale improves or until data improve. We really have to. We really have to also, inside own company data sets, put more negative results in there and put things into more of a common format, so it's easier to get them fed into the learning models. Not easy, though.
Totally understood, Derek. Yeah, I think we're on the same page here. So glad to have you here with your thoughts. Okay, so more questions. Hopefully we can hold on, Derek. There's still some time. So
I can go as long as you need.
Okay, that's awesome. All right, here's one with 6 likes. All right. Docking is used for structure-based prediction of protein-ligand interaction, as well as VS. Sorry, help me understand what that is.
Virtual synthesis, maybe?
Virtual screening.
Virtual screening, sorry.
Yeah. What's your opinion to apply the docking approach for AI ML?
Yeah, I think, virtual screening is the goal. That's what medicinal chemists really want out of computational techniques, is to rip through a gigantic list of compounds that we haven't made yet and tell us which ones to make. That gets back to the what compound should I make next problem that I mentioned. And I think I know what you mean by the doping approach to that. We don't have the horsepower to do this de novo, so we have to sneak up on it. And I think that's what you're talking about with the doping approach. You have to start somewhere, and you have to give the model something to chew on. That's what Schrodinger is talking about with running some experimental results and feeding those into your FEP calculations, so you at least have some ground truth to go from. I think anything that does not promise what I keep calling computational nirvana, that is doing this without recourse to any physical data, anything that does not promise that is, I think, in my mind, ranked higher than stuff that tries to do it all on a by your bootstraps technique. We're just going to look at these and tell you. You need some physical truth, and you need to bias your calculations, because otherwise the problem is too hard. It takes too much computing power. Look at the universe of small molecules. There's probably 10 to the 60th, 10 to the 62nd reasonable looking small molecules. That's a calculation from some years ago. Look at what we're doing now with all these bifunctional degraders and gigantic protein-protein interaction inhibitors. There's probably a lot more than 10 to the 62nd useful compounds out there. No computational method can attack that de novo. So yeah, you need all the help you can get and all the crutches that you can use along the way. And you have to be honest that that's what they are, and that's what you have to use.
Thank you, Derek. More questions coming in. So here's another one. It says, why do you think there are only about 3,000 approved drugs worldwide, USA, Europe, Japan, Korea? Can AI/ML be useful in drug repurposing?
Yeah, well, I mean, you say only 3,000 drugs. I'm surprised any drugs work at all. The longer I go in the industry, I'm just amazed and celebratory when stuff actually works. So I've been dealing with that high failure rate for so long. And drug repurposing is a tough one. And I know that there's been a lot of effort over in various parts of NIH on that. It's a seductive idea, but it is very difficult to realize in practice. The number of successful drug repurposing efforts, I think, can be counted on the fingers of one's hands, and maybe on the fingers of only one of those hands, depending on your definitions. It's very hard. Of course, that doesn't mean you shouldn't try it. Every time a new disease comes along or some new target, sure, run it through, because if you actually do find something, the promise is realized. That so much the groundwork has been done that you can advance very quickly. But your chances of being able to do that are so small. I mean, look at all the drug repurposing that went on as the coronavirus kicked in. And I don't count, for example, the steroid therapy. I don't count that as drug repurposing, because that's using the drug, the glucosteroid, that's using it for its intended purpose. It just turns out it was a lot more useful than we thought it would be for this particular virus. I'm talking about, finding something that's just a totally different drug from a totally different therapeutic area that, oh my God, happens to work in some part of the life cycle of this virus. So much of that stuff was tried, and so much of it just went down the drain. Drug repurposing is endlessly appealing, but it's a very wide desert to cross in practice. So I don't actually hold out too much hope of increasing the number of new drugs significantly through drug repurposing. I think we should be very happy when we can get it to work at all. Sorry to be a downer.
Thanks, Derek. All right, more questions. The questions are increasing as we move forward.
Yeah.
So there's a lot of questions, but maybe like a few more, and then we can stop here.
Up to
Okay.
you. Just.
Oh, I appreciate it, Derek. Maybe we'll go a little bit over time then.
Oh,
We
no
still have
problem.
3 minutes. Okay, great.
No problem. Keep on going.
Okay, so many thanks for this interesting talk. Another problem I see is the gender data gap. For the assays, it's probably rather a sex data gap. But regardless, the absence of data on female animals would extrapolate this gap further by training models on incomplete data sets. Is that right? Thank you.
I think that's a valid point, and it's part of a larger problem in that, I mean, look at our animal models in general. They are generally very rather defined, or maybe too well defined, inbred strains of particular rodents for the most part. And that can tell you a lot, but can only take you so far. That's a general reductionist problem. We've had to reduce the problem down to where we use, male C57 blacks. Yep, those are the mice we're using. Yep. And that's just because otherwise the problem gets too huge. But it's absolutely true that if you want a data set to feed into a machine learning model that you expect to tell you something interesting, you need much wider ranging stuff than that. And yes, the male-female difference in these has to be part of that. You think it has to be. There are so many biochemical differences along the way. I mean, just to pick one, look at the huge disparity in autoimmune disease in female versus male. Now we're starting to understand why that might be. You've seen some recent work on that with the gene silencing on the second X chromosome, but it just highlights the fact that there are a lot of very important differences there that we are probably missing. So yeah, I agree with that.
Thanks, Derek. Another question here, as you suggested, the limiting factor in high quality data, which I totally agree, is high quality data, which I totally agree. But collecting data piece by piece, like PDB, also takes decades.
Yes.
What's your opinion on building AI ML models to enhance human model interactions so that virtuous learning cycles can happen more efficiently? Thanks.
Yeah, that's a tough question, because you would hope that there's a way to do that, but I have to say I don't know quite how to do that, because you are coming perilously close to asking for a violation of a conservation law of knowledge and getting something for nothing. Frankly, that's what a lot of people expect from AI and ML. They want an oracle. They want to look over a data set and say, aha, here is the answer. Here's the rule. Here's the correlation that you hadn't known about. But if that correlation wasn't in the data, the model can't tell it to you. It's very good at finding correlations that you may have missed, but the answer had to be in there all along. I sound like the end of a bad movie. The answer had to be in there all along for the machine language or AI model to tease it out. It will not generate something from nothing. So that's the problem. But I agree. A database like the PDB is the result of a long amount of effort and work. The hope is that, for example, with, say, X-ray structures or cryo-EM in the PDB, that automation is increasing the number of submissions constantly. So we are going to have to recruit other machines to help us generate reliable data for the other set of machines. We need all the help we can get.
Makes sense. Thanks, Derek. There's a comment here. It's a lovely comment. It says, I would just like to say you have a very soothing presentation voice. And people have liked that. So maybe.
I like that. I've been told several times I need to do podcasting, but there are only so many hours in the day.
Ooh, that's a great idea, Derek.
Oh, damn it. It probably is. Stop telling me.
I would totally listen to that. I'm sure a lot of people would. All
maybe
right.
when I retire, who knows?
We have plenty of more questions, but we're on time. Would you like to go 5 more minutes to take a few more questions? Okay, let's do that. Here's a question that says 75% of ideas good medicinal chemists have the same intuition and come up with the same chemicals to make after HTS or SAR round. We should be able to model it. Agree with Derek 100% that the biological assays with predictive validity are the most important aspect for medicinal chemists. You know this, Serene, as well. The fact is that it's just hard to recapitulate the biology in a dish. So finding new medicines will be tough for some time to come. This is a colleague of mine, Sam Patnaik, medicinal chemist here, group lead at NCATS.
I agree with that. And it's true that medicinal chemists do tend to often come up with fairly similar ideas. I don't know if that's a bug or a feature, though. A lot of us have been burned in similar ways during our careers, but you do run into some outliers. You'll have some structures that go up on the board and there'll be one or two folks in the room who go, oh, no, no. And it turns out they worked on a project where those particular structures really betrayed them. And other people are like, no, that looks okay. And maybe they're right. Maybe these structures don't betray you all the time and they're worth a shot. Or maybe it's just because they haven't actually encountered the horribleness that these things are going to bring down on their heads. It's very hard. This gets into the argues about frequent hitters and pains compounds. And on the one hand, I think there really are pains. There really are frequent hitter type compounds that need to be avoided if you have any other choice. On the other hand, you can't just guarantee quality by running a pains program and ripping out all the bad stuff, because we don't know about all the bad stuff. And when you try to rip out all the bad stuff, you're going to rip out some good stuff too. It's harder than it looks.
Yeah, and that's, I think, like what Sam was also mentioning, the importance of assays.
Oh yeah.
When it comes to counter assays and all those things.
Ground truth. I mean, we never forget that, and always run an experiment if you can. Francis Crick used to talk about that. He would say, you know, the thing to do is just go ahead and try it. He said, theorists don't like this approach very much, but the way I've distilled that down is never talk yourself out of an easy experiment. Run the thing, see what happens.
Makes total sense. All right, one more question here. Do you think pharma has learned these same lessons about reductionist models when it comes to AI, or are they following the siren school again?
I think that there are many people in pharma who have learned those lessons, but whether pharma with a capital P has learned them or not, I kind of doubt it. And I think we're going to have to, I think people are going to continue with the way we're going because it is what's worked. And the only way that's going to persuade people to do things differently is when it really starts to stop working and got your back against the wall. So until that happens, I don't think there's going to be any gigantic revolution in the way pharma does research, but there are plenty of people inside pharma who've had these same thoughts, that's for sure.
I see. Thanks, Derek. Any thoughts on AI/ML use with clinical trial design to increase that 13%?
Yeah, I hope that it can help there. I've been surprised at how comparatively little Bayesian trial design we've seen over the years, because that, you know, you go back and you read the literature and it's always going to be one of these next big things that we're going to see a lot more of. And we do see some, but we don't see quite as much as I thought there would be. And it's a more complicated way to set up a trial and more labor intensive. But yeah, I wonder if there is a way to use these computational approaches to deliver more bang for the buck as far as more useful data per trial participant or dollars spent. I hope there is. I don't know enough about clinical trial design. I mean, I know a lot more than the average guy on the street, but I don't know enough about it, having not had to do it for a living, to tell you if that's possible or if I'm just being over optimistic, but I sure hope it's possible.
Thanks, Derek. Me too. Maybe one or two more questions, and that's it. Just like, we're so thankful. We're taking so much of your time, and
no problem.
This is great.
I can do this stuff for an extended period. It's a real problem for people who are within hearing range.
Okay, here's another question. What are the limitations of current AI/ML approaches in predicting protein dynamics And how might this be overcome in future developments?
Yeah, dynamic. That is a tough one, and I have a very cynical response to that, which I like to is trouble believing the static computational result just of one structure on the screen. How am I supposed to feel better if I'm looking at 30 of those a second going by as a movie? But on the other hand, it's obvious that the static structure on the screen, which is, of course, what you get from an X-ray or a cryo-EM or something like that, or most computations, is not the real story. Proteins move, and their movements are incredibly important. But I think it increases the computational problem drastically to try to deal with this. And I am not at all sure that we have the horsepower yet to deal with it in the way that some people seem to think, or in the way that a lot of us hope. So I think that adding that dimension of time and adding the dynamics to it with changes over time, picosecond by picosecond or even shorter, is going to be essential, but it just adds so much computational complexity. You'd hope that there will eventually turn out to be pattern matches similar to the ones we use for static structures that gave us AlphaFold and Rosetta Fold. There may be time-dependent pattern matches where you say, okay, if you're like this, generally this part goes this way and this part closes over this way. For the most part, it does that when you have this kind of structure. I hope that eventually there will be enough dynamic ground truth, which ain't easy to get, but enough dynamic ground truth to let us start pattern matching that way. But I'm not going to expect that next month.
Makes sense. All right. One more question. Could pharma be incentivized to share the overwhelming amount of data never published from drug discovery projects? Example, patent life extends for publishing all preclinical data associated with an approved drug.
That would be an incentive, and that's the sort of war club that you would have to use. Because I think if you're going to try to provide incentives for farmers to do that, it's going to have to be the equivalent of at gunpoint. And that's what you're describing is, messing with patent lifetimes is pretty much the equivalent of doing it at gunpoint from the IP standpoint, which is what the industry lives and dies by. So it would have to be something like that. And I agree, there's a tremendous amount of data in there, but don't get your hopes up too high because a lot of that data is in ugly shape. It was collected in a lot of different ways by a lot of different people, annotated and formatted all over the place in different ways. And that's what companies are finding as they do their data quality initiatives. It's you open up the vault, you're like, oh my God, look at this. How are we going to deal with this? So we're kind of aghast internally. And I think that you would be aghast on the outside if you got a good look at it. It's not like there's this gigantic well-lit basement of beautifully formatted data sitting there waiting for you. It's there, but it's going to need a lot of work.
Thanks, Derek. We're 10 minutes past time.
Okay.
So I'm going to stop. There's so many more questions. And I think maybe we can just host you back again and we keep
anytime.
talking. So this has been incredibly, insightful. We're extremely thankful for having you here. Everyone, if we didn't get to your questions today, hopefully maybe we can host him, Dr. Lowe back, and then we can continue talking.
Be glad
Derek,
to.
I appreciate the time you took to answer all these questions, the amazing insights, excess time you stayed over. We're really appreciative for you. Thank you again.
No problem. Enjoy
and
it.
everyone else. Thank you for being participating in this webinar series. We'll see you in this next quarter for the next talk. All right. Thank you, everyone.
Bye.
Have a good one. Bye.
Bye.